Yes, thank you, Fernando. I was honored to be asked to present more than 4 decades of Professor Marcello Barbieri's work. Last year, he generously summarized this intellectual journey in 12 key concepts published in Biosystems. Today, I will try to guide you through those ideas. The next one, please, Fernando. Nowadays, biology is largely organized around two dominant paradigms. The first is the idea that life is chemistry, that everything living can, in principle, be explained by physical and chemical interaction. The second is the idea that life is chemistry plus information, genetic sequences, regulatory networks, and computational descriptions. What I would like to present today begins with a third possibility, still a minority view, but one that I believe, and drawing closely on Marcello Barbieri's work, becomes unavoidable once we take experimental evidence seriously. This third paradigm states that life is chemistry, information, and codes. Over the past decades, biology has revealed the existence of hundreds of biological codes, operating at molecular, cellular, neural, and organismal levels. Despite this growing body of evidence, such codes remained largely absent from standard textbooks. What exists in nature, however, eventually reshapes our theories. When that happens, codes will no longer appear as metaphors or curiosities, but as part of the conceptual core of biology. The aim of this talk is therefore simple: to introduce the key concepts of this third paradigm, now known as code biology, and to show why it offers a coherent framework for understanding what makes life fundamentally different from matter. The next one, please, Fernando. Modern biology usually describes the cell as a system made of genotype and phenotype, a kind of biological computer, with genes as software and proteins as hardware. This picture is familiar, intuitive, and incomplete. When molecular biology began to study protein synthesis in detail, something decisive became clear. Genes and proteins do not arise spontaneously inside the cell. They are manufactured by a complex system of molecular machines built largely from RNA. In the early 80s, Marcello Barbieri proposed that this manufacturing system deserved a category of its own. Its components were called ribosoids, and the system as a whole was named the ribotype. From that point on, the cell could no longer be described as a dual system. It became a trinity: genotype, phenotype, and ribotype. At first sight, the ribotype may seem redundant, but it is fundamental. We can speak of genes and proteins in a biological sense only when a translation apparatus exists. Genotype and phenotype, therefore, do not exist independently. They exist because the ribotype exists. The translation machinery has both logical and evolutionary priority over them. Looking back to early life helps clarify this. Primitive systems already contained RNA, protein complexes performing gene-like and protein-like roles. Later evolution replaced ribogenes with genes and riboproteins with proteins, but the ribosoids of the ribotype were never replaced. They remained indispensable. Every living cell today contains a ribonucleoprotein system that manufactures proteins according to the genetic code. The self is therefore not best described as a computer, but as a code and template dependent protein maker. In short, a code maker. This is what turns the cell into a genuine trinity. Genotype as the seat of heredity, phenotype as the seat of metabolism, and ribotype as the seat of the genetic code, the system that makes biological meaning possible at the molecular level. Originally, this perspective was called semantic biology. As more biological codes were discovered, the name code biology became unavoidable because it is the existence of codes that makes meaning an objective feature of living systems. So, to say that the cell is made of genotype, phenotype, and ribotype is to say something very precise. The cell contains its own code maker, and for that reason, it is not a biological computer. This is the first key concept of code biology. Fernando, the next, please. The classical debate on evolutionary mechanisms crystallized in the 30s and 40s with the modern synthesis, which treated natural selection as the sole driver of evolutionary change. But this raises a simple question. Where does natural selection come from? At the molecular level, heredity depends on coping genes. Coping is not neutral. Repeated in time, in a world of limited resources, it inevitably generates variation and competition. In that sense, natural selection emerges from molecular coping under constraint. This would make natural selection the only evolutionary mechanism if coping were the only fundamental process of life. But biology tells us something else. The discovery of the genetic code showed that life is not based on copy alone. At the molecular level, life relies on two basic mechanisms: coping genes and coding proteins. If both are fundamental, then evolution must involve two distinct kinds of change. Barbieri proposed to call them evolution by natural selection based on coping and evolution by natural conventions based on coding. These two mechanisms are not merely different in degree, they are different in kind. Coping generates modified versions of existing molecules and therefore produces what we may call relative novelties. Coding works differently. By establishing correspondence between independent molecular worlds, new coding rules can create relationships that never existed before, what Barbieri described as absolute novelties. This is also a second crucial distinction. Changes in coping alter information. Changes in coding alter meaning. Information and meaning are not the same thing, and the contrast between coping and coding reflects precisely that difference. At this point, it is useful to be precise about what code biology means by meaning. In classical code biology, a code is a set of rules used by a system to generate a new world of objects from the information provided by a first world of objects. The objects of the second world did not exist before the code and therefore be regarded as the meaning of the objects of the first. A simple illustration is Morse code. The letter A is the meaning of dot dash. The same principle applies to all biological codes. We therefore arrive at a clear conclusion. Coping and coding are fundamentally different mechanisms of life, and for that reason, evolution itself must be understood as the result of two complementary processes: natural selection and natural conventions. This is the second key concept of code biology. Fernando, 3, please. Genes and proteins do not arise spontaneously. They are produced, assembled step by step by molecular machines. This simple fact has a powerful consequence. Genes and proteins are manufactured molecules. In other words, molecular artifacts. Once this is recognized, a broader conclusion follows. If genes and proteins are manufactured, then all biological structures are manufactured. Life as a whole is not a collection of spontaneous objects, but a process of artifact making. Molecular biology gives us clear reasons for this view. The production of genes and proteins requires more than catalysts. It also requires templates, structures that specify the order in which components are assembled and thereby determine biological form. Here, we encounter a fundamental difference between chemistry and biology. In chemical objects, order arises from within the molecule, from internal physical constraints. In biological objects, order is imposed from the outside by templates guiding assembly. If we now compare gene and protein production, another decisive distinction appears. Both rely on catalysts and templates, but protein synthesis requires something more: coding rules. Genes can be produced by copying templates. Proteins cannot. Their sequences must be translated from nucleic acids into amino acids through a code implemented by molecular adapters, such as transfer RNAs. We, therefore, arrive at a clear conclusion. At the basis of life are two distinct manufacturing processes: the copying of genes and the coding of proteins. Both are indispensable. From this perspective, the origin of life can be described precisely. It was the origin of the first systems capable of manufacturing genes and proteins by copying and coding. This idea of artifact making also resolves a classic problem. Matter consists of spontaneous objects. Life consists of manufactured ones, that is, molecular artifacts built by molecular machines. The transition from chemistry to biology was therefore not miraculous, but natural. The appearance of systems capable of building artifacts. Life began when molecular machines started populating the primitive Earth with manufactured molecules, and this is the third key concept of code biology. Fernando, the next one, please. Life is clearly different from matter. But how deep is that difference? Is life simply an extremely complex form of chemistry, or is something fundamentally new? To answer this, we first need to clarify what a fundamental difference means. Physics offers a useful lesson. Every scientific field begins by identifying what it must describe: its basic quantities. Space, time, mass, and electric charge are not explanations. They are the core features that define entire domains of phenomena. Take astronomy. If planetary motion is described only in terms of space and time, we can build either a Ptolemaic or a Copernican model, introduce mass, and an entirely new framework becomes possible. Newtonian mechanics and universal gravitation in general. New scientific worlds emerge when new fundamental variables are discovered. So, the key question becomes unavoidable. Does life require new fundamental variables? Here, the answer is clear. Living system cannot be described without genetic sequences, forms of organic information, and coding rules, forms of organic meaning. These are indispensable and cannot be reduced to physical or chemical quantities. They also display two striking properties. A change in a single element of a biological sequence can generate an object with entirely new properties, even though the sequence functions as an integrated whole. And if we know the first n elements of such a sequence, we cannot predict the n plus 1. Biological sequences cannot be derived from simple descriptions. They must be specified in their natural order. For this reason, genetic sequences and coding rules are not computable in the usual sense. They must be listed and preserved. This stands in sharp contrast with physics and chemistry, where many quantities can be derived from equations. We therefore arrive at a clear conclusion. Just as mechanics, electromagnetism, and particle physics form distinct domains because they rely on different basic variables, biology is also a distinct domain. Life is fundamentally different from matter because it requires new foundational features in addition to those of physics and chemistry, organic information and organic meaning. And this is the fourth key concept of code biology. Fernando, please, another one. One of the most striking facts in biology is the stability of the genetic code. Since its origin nearly 4 billion years ago, it has remained virtually unchanged. This stability is often explained by calling the code a set of constraints, but that is not enough. The genes implementing the code evolve like all others. They mutate and drift. And yet, the code itself has been preserved with remarkable fidelity. Its conservation, therefore, cannot be passive. It must depend on a biological mechanism that actively restores the code. To understand this, we need to look at life before and after the genetic code. Early protein synthesizing systems explored many coding possibilities. Coding rules were fluid and evolving. After the genetic code emerged, the situation changed dramatically. Further modifications were no longer tolerated. The apparatus became a code-conserving system, even while other parts of the cell continued to explore new codes. This dual organization already existed in the ancestors of eukaryotic cells. Systems devoted to conserving the genetic code alongside others capable of generating additional ones. It tells us something fundamental. The origin of life depended on the ability to generate coding rules. The subsequent evolution of life depended on two complementary processes: conserving existing codes and creating new ones. Together, this forms a deeper biological dynamic that Barbieri called code poiesis. It is useful to contrast this with autopoiesis. Living cells are often described as self-fabricating systems, but before the genetic code existed, early systems were producing descendants that differed from themselves. They were not autopoietic in the strict sense, yet they were evolving coding rules. They were, in short, code poietic. The conclusion is striking. Autopoiesis is powerful, but not fundamental. Code poiesis precedes it. The capacity to generate, conserve, and expand codes is the deeper process underlying the origin and evolution of life. And this is the fifth key concept of code biology. Fernando, another one, please. For a long time, the tree of life was reconstructed by comparing individual molecules across species. With whole genome analysis, however, a clear picture emerged. Modern eukaryotes descend from a small number of lineages radiating from a common ancestor. Cellular evolution was shaped by two major events: the emergence of the last universal common ancestor, LUCA, around 3.5 billion years ago, and roughly 2 billion years later, the last eukaryotic common ancestor, LICA. The long interval between LUCA and LICA was not merely a period of accumulating components. It was marked by the appearance of many new biological codes. During eukaryotic evolution, systems such as splicing, signaling, cytoskeletal and nuclear codes, ubiquitin codes, and others emerged. What matters here is not the list, but the pattern. A striking contrast then becomes visible. Prokaryotes appeared early in Earth history and largely conserved their level of complexity, as shown by ancient stromatolites resembling modern cyanobacteria. Eukaryotes did the opposite. They repeatedly increased their complexity and eventually crossed the cellular boundary into multicellular life. Code biology offers an expected explanation. The decisive factor is not size, energy, or metabolism, but the number of codes. Prokaryotes evolved relatively few. Eukaryotes evolved many more. As new codes appeared, new organizational possibilities became available. The conclusion is straightforward. Biological complexity is closely associated with the number of codes a system possesses. Codes expand the space of possible forms, functions, and interactions. The long history of eukaryotic evolution can therefore be understood as a history of a code expansion. And this is the sixth key concept of code biology. Fernando, another one, please. The number 7. For almost 3 billion years, life on Earth existed only as single cells. Evolution refined cellular organization, but life remained confined within the cell. Eventually, some eukaryotes crossed the threshold and gave rise to multicellular organisms, triggering a new wave of biological innovation. Multicellularity was not simply a matter of having more cells. It required new ways of coordinating, differentiating, and integrating cellular activity. Once again, code biology points to the same mechanism: the emergence of new biological codes. With multicellular life came developmental codes, such as the OX code for body plans, along with adhesion, transcriptional and histone codes, apoptosis codes, and bioelectric codes guiding growth and regeneration. The list is long, but the pattern is clear. Each new level of organization required new rules of correspondence. With the origin of animals, evolution took another decisive turn. Neural activity, instincts and feelings appeared, and life entered a new domain structured by codes. Neuroscience has revealed sensory codes, synaptic codes, and spatial codes in the hippocampus. One discovery is especially illustrative. The hippocampal space code, recognized with the Nobel Prize in Medicine in 2014, showed that spatial navigation is organized according to discrete coding rules. Across all levels, the message is consistent. Whenever life crosses a new threshold of complexity, from cells to multicellular organisms, and from tissues to nervous systems, it does so by introducing new codes. The origin of multicellular life was therefore not merely an increase in size or number, but the emergence of a rich landscape of new codes that enabled higher levels of organization. This is the seventh key concept of code biology. Fernando, number 8, please. There is a strong evidence that virtually all animals share the same basic feelings and instincts: hunger and thirst, fear and aggression, reproductive drives and responses to light, sound, pressure, and temperature. The remarkable universality suggests that they appeared in a common ancestor and have been inherent ever since. Animals who share another striking feature. Sensory organs deliver a limited set of inputs to the brain, and the brain produces a restricted set of outputs. Basic feelings, instincts, and action tendencies. Between them lies a complex neural machinery. Crucially, experimental evidence shows that the mapping between inputs and outputs is not fixed by physics and chemistry alone. It must be established by biologically arbitrary rules, in short, by a neural code. This leads to what Barbieri called the code theory of mind. The proposal that there exists a universal neural code at the origin of mind, just as there is a universal genetic code at the origin of life. Within this framework, feelings and instincts are not accidental byproducts of neural wiring, computational outputs, or vague emergent properties. They are manufactured. Just as proteins are assembled from amino acids according to the genetic code, feelings and instincts are assembled by neural adapters according to a universal neural code. From this perspective, they are not mysterious inner states, but brain artifacts produced in a lawful, rule-governed manner, and this provides a parsimonious explanation for why the same basic feelings and instincts are found across virtually all animals. This is the 8th key concept of code biology. Another one, Fernando, please. Animals can give meaning to the environmental signals through neural codes. But some animals have evolved something more: the ability to interpret what is happening in the world. The difference can be illustrated with a simple example. When a snake loses sight of its prey, it stops. When a wolf loses sight of its prey, it continues the chase. The difference is not one of intelligence, but of mechanism. The snake follows fixed coding rules. The wolf performs an act of interpretation. In doing so, the wolf makes a mental leap behind what is immediately given, what Charles Peirce called abduction, a plausible guess that goes beyond available data. Animals, therefore, evolved two forms of semiosis. One based on codes, operating through fixed correspondences, and another based on interpretation, operating through abductive reasoning. Interpretation is not universal in life. It appears only when neural systems reach higher levels of complexity. A similar pattern appears in artificial systems. Only sufficiently complex neural networks show abductive-like behavior. Simpler systems merely execute rules. This reinforces a central point. Interpretation is not primitive. It is a late evolutionary achievement. This brings us to a classical debate in biosemiotics. Thomas Sebeok argued that the origin of life was the origin of semiosis and therefore of interpretation. Code biology accepts part of this claim, but not all of it. Codes can produce semiosis, and life began with the genetic code, but the first living systems were not interpreters. Interpretation evolved much later, with complex animals, long after the origin of life. The crucial distinction is therefore simple. Codes are primordial. Interpretation is derived. Life began with coding. Interpretation came later. This is the ninth key concept of code biology. Fernando, another one, please. Living organisms have often been described as self-fabricating systems, autopoietic systems that continuously produce their own components. This idea has been developed in different ways, from von Neumann's self-reproducing automata to the theories of Maturana and Varela and Robert Rosen's metabolism repair models. Rosen, in particular, argued that organisms persist by acting in the present to avoid harmful future states, and therefore defined living beings as anticipatory systems. If that were true, the origin of life would coincide with the origin of anticipation. But at closer look at biology, biology suggests otherwise. Some animals clearly anticipate future conditions, such as storing food for scarcity. Others do not. Primitive organisms like sponges and sea anemones react to present stimuli but do not act in anticipation. This simple contrast has an important consequence. Anticipation cannot be universal in life. It must be an evolutionary acquisition that appeared only in certain lineages alongside increased neural and behavioral complexity. So, while anticipation is a powerful biological capacity, it is not primordial. It did not exist at the origin of life. It evolved later in animals with more complex forms of organization and control. And this is the 10th key concept of code biology. Fernando, 11, please. Thank you. Feelings and instincts are first-person experiences. They give us the sense that we inhabit our body and live a personal life. They are profoundly private. No one else can feel my pain. Yet they are natural processes that call for biological explanation. Consider a simple example. If you injure your toe, signals travel to the brain processes them, and commands return to the muscles. In that description, the brain is the observer and the toe is the observed. Yet something extraordinary happens. Although pain is generated in the brain, it is felt in the toe. Observer and observed collapse into one. The experience is displaced from where it is produced to where it is lived. This is not an illusion. The brain actively transforms signals, correcting retinal motion, compensating for changes in light, and stabilizing perceived size and color. First-person experience arises from this same capacity. Highly differentiated neural systems create a functional short circuit between brain and body. This was a genuine biological revolution. Primitive nervous systems were transformed into conscious brains, and something entirely new appeared in the nature: subjectivity. With that, the world acquired a first-person dimension. The origin of first-person experience was the origin of consciousness. This is the 11th key concept of code biology. Another one, Fernando, please. A special and highly distinctive form of semiosis is language. The capacity to give meaning to words. Language does not arise spontaneously in an isolated brain. It is produced through a process of languaging that requires prolonged interaction between human beings. In this sense, language is irreducibly social. This becomes clear in dramatic cases. Children who grow up without sustained human contact, the so-called wolf children, almost completely lose the capacity to acquire language. A newborn can develop language only if postnatal development is actively supported by others. So, language is also unique to our species. One possible reason lies in a peculiarity of human development. Humans are born biologically premature, with extended postnatal dependence that makes vocal interaction one of the primary ways of engaging caregivers. Through this prolonged interaction, infants gradually associate sounds with situations, objects, and eventually names. In this way, a child builds not only a world of things, but also a world of words. Thomas Sebeok described this symbolic layer as a modeling system, a level of organization that restructures experience itself and makes language possible. With language, semiosis reaches a new stage. Meaning is no longer produced only by codes or neural processes, but also by shared symbolic conventions. And this is the 12th and the last key concept of code biology. Fernando, the last one, please, the conclusion. For a long time, codes were thought to exist only in human culture, in language, symbols, and social conventions. Biology, on that view, was chemistry, or perhaps chemistry plus information. What we have seen today suggests a very different picture. Experimental biology has revealed codes at every level of life, at the cellular level in organic codes, at the multicellular level in developmental and embryonic codes, and at the neural level in systems that organize feelings, instincts, and behavior. Living systems appear to use hundreds of such codes, but the importance of this fact goes far beyond counting them. It forces us to rethink evolution, not as a driven by natural selection alone, but as the result of two complementary mechanisms: natural selection and natural conventions. It reshapes our understanding of life's origin, not as the emergence of spontaneous objects, but as the emergence of artifact-making systems. And it invites us to rethink mind and meaning, not as byproducts of complexity, but as outcomes of coding processes that appeared at different moments in evolutionary history. We have followed this path through 12 key concepts of code biology. Taken together, they form a coherent framework for understanding what makes life distinctive. The central claim is simple but far-reaching. Life is chemistry, information, and meaning, because codes produce meaning in the strict sense proposed by code biology. Meanings are the objects generated by codes. If this is right, then codes are not marginal curiosities. They are part of the very fabric of life. And the last one, please, Fernando. Thank you very much for your attention. And I warmly invite you to continue the discussion on our collaborative platform, codebiology.net, where experts from different fields engage with questions about code biology. Thank you. Thank you very much.